Bioresources and uses of biodiversity. Maximized word limit, JRF level, paragraph format, based on CSIR-NET Life Sciences previous year question areas, note tables, with 2 to 3 solved PYQs. Biodiversity, or biological diversity, encompasses the variety and variability of all forms of life on Earth, including genetic, species, and ecosystem diversity. Bioresources are the components of biodiversity that hold actual or potential utility to humans, such as plants, animals, microbes, and their products. The study and application of bioresources, a core area in applied biology, has profound implications for medicine, agriculture, biotechnology, and sustainable development. The Convention on Biological Diversity (CBD) defines bioresources as genetic resources, organisms or parts thereof, populations, or any other biotic component of ecosystems with actual or potential use or value for humanity. The uses of biodiversity can be broadly categorized into direct, indirect, and option future potential uses. Direct uses involve tangible biological products like food grains, vegetables, fruits, fish fiber, cotton, jute, coir, fuel wood, junk medicines, quinine, artemisinin, taxol, and industrial products, rubber, resins, oils. Indirect uses relate to ecosystem services such as pollination, nutrient cycling, climate regulation, water purification, and soil formation. Biodiversity also underpins cultural, spiritual, and recreational benefits. Option values reflect the future, as yet unknown, uses of biodiversity, which may be critical for medical or technological innovations. Bioresources in agriculture include crop wild relatives and land races that carry genes for resistance to pests, diseases, or environmental stress. These are crucial in crop improvement programs via plant breeding or genetic engineering. For example, wild varieties of rice and wheat have contributed genes for drought and salinity tolerance in modern cultivars. Similarly, microbial bioresources are exploited for nitrogen fixation, e.g., rhizobium, azospirillum phosphate solubilization, Bacillus, Pseudomonas, and biopsicide development, Bacillus thuringiensis. In pharmaceutical sciences, over 25% of drugs used in modern medicine are derived from higher plants, many of which are from tropical rainforests. Examples include morphine from Papaver somniferum, aspirin from Salix alba, and vincristine from Catharanthus roseus. Additionally, marine bioresources, such as sponges, corals, and cyanobacteria, were rich sources of novel anti-cancer and anti-inflammatory compounds. Biodiversity also fuels biotechnological innovation. Enzymes from extremophiles like Thermus aquaticus, source of Taq polymerase used in PCR, or Pyrococcus furiosus, used in high-temperature industrial processes, illustrate how genetic diversity translates into technological breakthroughs. Metagenomics allows access to genetic resources of uncultivable microbes from diverse environments, enabling novel enzyme and metabolite discovery. Genetic resources are protected under international treaties like the Nagoya Protocol, which governs the access and fair benefit sharing of genetic material. The International Treaty on Plant Genetic Resources for Food and Agriculture (ITPGRFA) ensures farmers' rights and conservation of crop diversity. In India, which is one of the 17 megadiverse countries, bioresource management is governed by the Biological Diversity Act, 2002, implemented through the National Biodiversity Authority (NBA). This act ensures sustainable use, conservation, and equitable benefit sharing. Traditional knowledge associated with biodiversity is documented in the Traditional Knowledge Digital Library (TKDL) to prevent biopiracy, as in the famous case where the U.S. patent on turmeric's wound healing properties was revoked. A key concern is the erosion of biodiversity, driven by habitat destruction, pollution, invasive species, overexploitation, and climate change. Loss of biodiversity not only reduces ecological resilience but also deprives humanity of potential bioresources, which could otherwise address future health, food, and climate-related challenges. Biodiversity is also being economically evaluated using models like Total Economic Value (TEV), which includes direct use, indirect use, and non-use values, existence and bequest values. Modern conservation approaches like bioprospecting, searching for valuable genetic and biochemical resources, community-based conservation, and ecosystem services valuation are integrating science with economics and ethics. In conclusion, bioresources are an integral part of biodiversity with immense actual and potential value. Their responsible utilization, equitable sharing, and long-term conservation are essential for human well-being, sustainable development, and ecological balance. For CSIR-NET Life Sciences, this topic is critical because it integrates ecology, biotechnology, policy, and ethics, areas that are often cross-linked in multiple sections of the exam. Previous year CSIR-NET questions with answers, CSIR-NET June 2016. Which of the following is a correct example of indirect use of biodiversity? A medicinal plants. Bee firewood collection, C pollination by bees, D collection of wild fruits. Answer, C pollination by bees. Explanation, this is an ecosystem service, not a direct harvest of biodiversity. CSIR-NET Deck 2019. The Indian law that governs access to biological resources and associated knowledge is Environment Protection Act, 1986. Bee Forest Conservation Act, 1980. C Biological Diversity Act, 2002. D Wildlife Protection Act, 1972. Answer, C Biological Diversity Act, 2002. CSIR-NET June 2022. Which of the following is a correct statement regarding bioresources? A. They are restricted to genetically modified organisms. B. They include only plants used for agriculture. C. They include all biotic components with potential utility. D. They are limited to medicinal plants and microbes. Answer, C. They include all biotic components with potential utility. Let me know the next topic in applied biology or any unit from the CSIR-NET Life Sciences syllabus. I'll continue preparing JRF level theory notes accordingly. You said, breeding in plants. ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on breeding in plants. Maximum word limit, JRF level, CSIR-NET focused, previous year question based, paragraph format, note tables. Plant breeding is the art and science of improving the genetic makeup of plants to enhance desirable traits such as yield, quality, disease resistance, and stress tolerance. This discipline, foundational to agriculture and food security, uses principles of genetics, reproductive biology, molecular biology, and biotechnology. Traditional plant breeding relied on phenotypic selection and hybridization, while modern approaches incorporate marker-assisted selection, tissue culture, genetic engineering, and genomic tools. CSIR-NET Life Sciences often tests conceptual understanding, breeding strategies, and integration of molecular tools with classical genetics in plant breeding. The process of plant breeding begins with germplasm collection and evaluation. Germplasm refers to the genetic material available for breeding, encompassing land races, wild relatives, traditional cultivars, and mutant lines. The variability present in germplasm is the foundation of breeding efforts. Following evaluation for desirable traits, breeders use selection, either mass selection selecting many plants with desired traits, or pure line selection selecting and propagating progeny from a single superior individual to improve populations. Hybridization is the crossing of genetically distinct parents to combine favorable alleles. The resulting F1 hybrids often exhibit heterosis or hybrid vigor, showing superior qualities like higher yield or stress resistance. However, in self-pollinated crops like wheat or rice, hybrid vigor tends to break down in subsequent generations, so selection must follow hybridization to fix desirable traits. For cross-pollinated crops e.g., maize, composite and synthetic varieties are developed through recurrent selection to maintain heterozygosity. Breeding for disease and pest resistance uses gene-for-gene interactions and relies on the identification of resistance R genes in wild relatives. Classical backcrossing introduces a resistance gene into a high-yielding cultivar, while retaining agronomic traits. In the Green Revolution, for instance, IR8 rice was developed through hybridization and selection for dwarf stature and high yield. Mutation breeding, induced by chemicals e.g., EMS or physical agents e.g., gamma rays creates novel alleles that are then selected for desirable traits. This has been successful in crops like groundnut and barley. However, the mutations are random and require extensive screening. The advent of molecular breeding revolutionized plant breeding. Marker-assisted selection (MAS) uses molecular markers RFLP, SSR, SNP linked to traits of interest to select desirable plants even in the absence of phenotype. This is particularly useful for traits that are difficult to screen phenotypically, such as root architecture or drought tolerance. The integration of QTL mapping allows breeders to identify genomic regions associated with quantitative traits, essential for complex trait breeding. Genomic selection (GS) is an advanced approach where genome-wide markers predict the breeding value of individuals, increasing selection accuracy and reducing breeding cycles. This is especially relevant in perennials or long gestation crops like coconut or oil palm. Transgenic breeding introduces foreign genes e.g., Bt, herbicide resistance into crops to improve resistance and quality. However, regulatory hurdles and biosafety concerns limit its widespread use. Genome editing, particularly using CRISPR, SOS9, is a frontier in plant breeding. It allows precise edits in endogenous genes to improve traits like flowering time, nutrient efficiency, and stress tolerance without introducing foreign DNA, making the plants non-transgenic in some regulatory contexts. Breeding also addresses abiotic stress resistance, an emerging priority in the face of climate change. Traits like drought tolerance, salt resistance, and temperature resilience are polygenic and complex. The identification of transcription factors e.g., DREB, NAC, and stress-inducible genes has facilitated molecular breeding for such traits. Plant breeders must also consider intellectual property rights (IPR) and farmers' rights, especially in countries like India under the Protection of Plant Varieties and Farmers' Rights (PPV and FR) Act. The balance between innovation and equity is crucial for sustainable breeding systems. In conclusion, plant breeding is a dynamic field integrating classical genetics, molecular tools, and biotechnological innovations. For CSIR-NET aspirants, understanding the methodologies, principles, and applications of plant breeding, particularly hybridization, marker-assisted selection, QTL mapping, mutation breeding, and genome editing, is essential. Questions often test your ability to connect genetics with applied breeding outcomes and understand trait inheritance under various breeding schemes. Previous year CSIR-NET questions with answers, CSIR-NET Deck 2016. Which of the following is an advantage of marker-assisted selection, MAS, in plant breeding? It allows naturalization of desired traits. B. It eliminates the need for hybridization. C. It allows selection of plants based on molecular markers linked to traits. D. It increases heterozygosity in crops. Answer. C. It allows selection of plants based on molecular markers linked to traits. CSIR-NET June 2020. Backcross breeding is generally used to A. Generate heterozygous lines. B. Introduce a single gene into a superior variety. C. Create transgenic plants. D. Develop polyploid varieties. Answer. B. Introduce a single gene into a superior variety. CSIR-NET Deck 2017. In marker-assisted selection, the use of molecular markers is advantageous because they are always dominant markers. B. They eliminate the need for any phenotype evaluation. C. They can help in selecting traits not expressed early in development. D. They are only applicable in mutation breeding. Answer. C. They can help in selecting traits not expressed early in development. Let me know the next topic you'd like detailed. We can continue building your full applied biology coverage or move to another unit of the CSIR-NET Life Sciences syllabus. You said, and animals, ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on breeding in animals. High quality, maximum word limit, paragraph format, CSIR-NET Life Sciences focused, with previous year questions and answers. Animal breeding refers to the controlled propagation of domesticated animals to improve desirable traits such as milk production, meat quality, egg-laying capacity, disease resistance, fertility, and growth rate. It forms a critical pillar in animal husbandry and sustainable livestock development. In the context of CSIR-NET JRF Life Sciences, questions focus on classical and modern genetic principles applied to animal breeding, inheritance of quantitative traits, artificial selection strategies, and integration of molecular tools like marker-assisted selection and genomic selection. The foundation of animal breeding lies in population genetics and quantitative genetics. Traits of economic importance in animals are usually polygenic, influenced by multiple genes, and show continuous variation, making quantitative trait loci (QTL) mapping and heritability estimation essential components of animal breeding programs. Selection intensity and response to selection depend on the heritability of traits in the selection differential, as explained by the Breeder's equation, R equals H squared times S, where R is the response, H squared is heritability, and S is selection differential. Inbreeding and outbreeding are two principal strategies in classical breeding. Inbreeding involves mating between related individuals to concentrate desirable alleles but increases the risk of inbreeding depression, where deleterious recessive alleles become homozygous. This is sometimes strategically used to fix desirable traits in pure lines, such as in laboratory model organisms like mice. Outbreeding, including crossbreeding, outcrossing, and species hybridization, aims to enhance heterozygosity and exploit heterosis, hybrid vigor, leading to improved performance in F1 individuals. For example, crossbreeding of Bos indicus Indian breeds with Bos taurus exotic breeds has enhanced milk productivity in cattle under Indian climatic conditions. Artificial insemination (AI) is a pivotal tool in modern animal breeding, allowing for genetic dissemination from elite males across large populations, even internationally. It reduces disease transmission and improves selection accuracy. AI forms the backbone of progeny testing, wherein the performance of offspring is evaluated to determine the genetic merit of breeding sires. This is a common practice in dairy cattle improvement programs. Open nucleus breeding systems (ONBS) represent structured breeding strategies where superior animals are maintained in a nucleus herd, and genetic gains are disseminated to commercial herds. This hierarchical model helps maintain genetic progress and control in large-scale programs. The integration of molecular markers in animal breeding has revolutionized selection accuracy. Marker-assisted selection (MAS) uses DNA markers, e.g., SNPs, microsatellites tightly linked to QTLs or major genes. High quality, maximum word limit, paragraph format, CSIR Net Life Sciences focused, with previous year questions and answers. Animal breeding refers to the controlled propagation of domesticated animals to improve desirable traits such as milk production, meat quality, egg-laying capacity, disease resistance, fertility, and growth rate. It forms a critical pillar in animal husbandry and sustainable livestock development. In the context of CSIR Net JRF Life Sciences, questions focus on classical and modern genetic principles applied to animal breeding, inheritance of quantitative traits, artificial selection strategies, and integration of molecular tools like marker-assisted selection and genomic selection. The foundation of animal breeding lies in population genetics and quantitative genetics. Traits of economic importance in animals are usually polygenic, influenced by multiple genes, and show continuous variation, making quantitative trait loci (QTL) mapping and heritability estimation essential components of animal breeding programs. Selection intensity and response to selection depend on the heritability of traits in the selection differential, as explained by the breeder's equation, R equals H squared times S, where R is the response, H squared is heritability, and S is selection differential. Inbreeding and outbreeding are two principal strategies in classical breeding. Inbreeding involves mating between related individuals to concentrate desirable alleles but increases the risk of inbreeding depression, where deleterious recessive alleles become homozygous. This is sometimes strategically used to fix desirable traits in pure lines, such as in laboratory model organisms like mice. Outbreeding, including crossbreeding, outcrossing, and species hybridization, aims to enhance heterozygosity and exploit heterosis, hybrid vigor, leading to improved performance in F1 individuals. For example, crossbreeding of Bos indicus Indian breeds with Bos taurus exotic breeds has enhanced milk productivity in cattle under Indian climatic conditions. Artificial insemination (AI) is a pivotal tool in modern animal breeding, allowing for genetic dissemination from elite males across large populations, even internationally. It reduces disease transmission and improves selection accuracy. AI forms the backbone of progeny testing, wherein the performance of offspring is evaluated to determine the genetic merit of breeding sires. This is a common practice in dairy cattle improvement programs. Open nucleus breeding systems (ONBS) represent structured breeding strategies where superior animals are maintained in a nucleus herd, and genetic gains are disseminated to commercial herds. This hierarchical model helps maintain genetic progress and control in large-scale programs. The integration of molecular markers in animal breeding has revolutionized selection accuracy. Marker-assisted selection (MAS) uses DNA markers, e.g., SNPs, microsatellites tightly linked to QTLs or major genes. For instance, selection for disease resistance in poultry against Marek's disease or improved litter size in pigs is facilitated by MAS. However, many traits are influenced by numerous loci with small effects, requiring genomic selection strategies. Genomic selection (GS) involves whole genome SNP profiling and statistical modeling to estimate genomic estimated breeding values (GABVs) without waiting for phenotypic data. It has reduced generation intervals and increased accuracy, especially in cattle and pigs. Genome-wide association studies (GWAS) identify genomic regions associated with traits, enabling finer genetic dissection of complex traits. Imputation techniques help reduce genotyping costs by using reference panels to predict untyped genotypes in breeding populations. Reproductive technologies like embryo transfer, in vitro fertilization (IVF), and cloning (e.g., somatic cell nuclear transfer (SCNT) are used to propagate elite genotypes. Cloning allows duplication of high-yielding animals, but ethical concerns, reduced lifespan, and abnormal development limit its commercial application. With the advent of CRISPR, Cas9 and other gene editing tools, animal breeding is now entering a precision era. Targeted genome editing has created disease-resistant pigs, e.g., PRRS-resistant, and polled cattle hornless, avoiding dehorning. These technologies promise rapid genetic improvement but raise regulatory, ethical, and consumer acceptance issues. In India, government schemes like National Artificial Insemination Program (NAIP) and Rashtriya Gokul Mission aim to improve indigenous breeds and promote sustainable livestock production. Regulatory frameworks such as breed societies, performance recording systems, and genetic evaluation reports are part of structured breeding programs. In conclusion, animal breeding integrates classical genetic approaches, reproductive technologies, and cutting-edge genomics to improve animal productivity, health, and welfare. For CSIR Net JRF aspirants, mastering these concepts, including genetic gain, selection indices, inbreeding coefficient, heritability estimation, and applications of MAS and GS, is critical to tackle applied biology questions effectively. Previous year CSIR Net questions with answers, CSIR Net December 2015. What is the main advantage of genomic selection over marker-assisted selection in animal breeding? A genomic selection uses fewer markers. Genomic selection does not require genotyping. C. Genomic selection predicts breeding value using genome-wide markers. D. Genomic selection is less accurate than MAS. Answer. C. Genomic selection predicts breeding value using genome-wide markers. CSIR Net June 2017. In animal breeding programs, which of the following strategies is commonly used to reduce the risk of inbreeding depression? A. Artificial insemination with elite sires only. B. Inbreeding within small isolated populations. C. Crossbreeding between unrelated breeds. D. Selection based on phenotype only. Answer. C. Crossbreeding between unrelated breeds. CSIR Net Deck 2019. Which of the following statements regarding marker-assisted selection (MAS) in livestock is true? A. MAS is only useful for monogenic traits. B. MAS cannot be used for traits with low heritability. C. MAS enables early selection before trait expression. D. MAS increases phenotypic variability. Answer. C. MAS enables early selection before trait expression. Let me know the next topic, or if you'd like to proceed to another unit in the CSIR Net Life Sciences syllabus. You said. Including marker-assisted selection. ChatGPT said. Here's the extended CSIR Net JRF Life Sciences lecture on breeding in animals, including marker-assisted selection, maximum word limit, paragraph format, highly exam-focused, with previous year questions. Animal breeding is the science of genetically improving livestock by selecting individuals with superior traits to produce offspring with enhanced performance. It combines traditional methods like selective mating and crossbreeding with modern molecular tools. The fundamental goal is to improve economic traits such as growth rate, disease resistance, reproductive efficiency, and product quality, milk, meat, eggs, wool, etc. In the CSIR-NET life sciences context, this topic merges quantitative genetics with biotechnology, making it a high-yield area for applied biology questions. Classically, animal breeding relied on phenotypic selection and pedigree-based selection, where breeders identified superior animals based on observable characteristics or family history. However, this approach has limitations, especially for traits with low heritability or those expressed late in life, e.g., fertility or longevity. To overcome this, estimated breeding values (EBVs) are calculated using statistical models like BLUV, best linear unbiased prediction incorporating phenotypic, pedigree, and environmental data to predict genetic merit. These values are used in progeny testing, especially in dairy cattle, where the genetic worth of sires is inferred from the performance of their daughters. The introduction of molecular markers marked a paradigm shift in breeding strategies. A molecular marker is a DNA sequence associated with a trait of interest and can be used to track the inheritance of that trait across generations. In marker-assisted selection, MAS breeders use DNA markers linked to quantitative trait loci (QTLs) to select animals carrying favorable alleles. In marker-assisted selection, MAS breeders use DNA markers linked to quantitative trait loci (QTLs) to select animals carrying favorable alleles, even before the traits are expressed. This is especially useful for traits difficult to measure or those with low heritability, like disease resistance or fertility. MAS typically involves three main steps: 1. Identification of molecular markers associated with the trait, e.g., SNPs, SSRs; 2. Genotyping of animals for those markers; and 3. Selection of individuals carrying desired alleles. For instance, in pigs, markers linked to the RYR1 gene help eliminate the allele responsible for porcine stress syndrome. In cattle, markers linked to DGAT1 gene are used for improved milk fat content. MAS reduces generation intervals and increases the precision of selection, especially when phenotypic data is unavailable or expensive to obtain. While MAS is effective for oligogenic traits controlled by a few major genes, its power is limited for polygenic traits, where the genetic basis is spread across many loci. To overcome this, genomic selection (GS) is employed. GS uses genome-wide SNP data to estimate genomic estimated breeding values (GEBVs) using statistical models like GBLUP or Bayesian approaches. Unlike MAS, GS captures the cumulative effect of thousands of loci, making it superior for traits like milk yield, carcass quality, or disease resistance. Examples of MAS in animal breeding include selection for scrapie resistance in sheep using the PRNP gene marker, selection against bovine leukocyte adhesion deficiency (BLAD) in cattle using DNA testing, use of FECB gene marker in sheep to improve fecundity. These programs have demonstrated increased accuracy, reduced costs, and shortened selection cycles. Breeding strategies also include inbreeding, line breeding, and crossbreeding. While inbreeding increases homozygosity and can fix desirable traits, it risks inbreeding depression, which reduces fitness. Hence, controlled outcrossing is often used to maintain genetic diversity. Crossbreeding combines breeds with complementary traits, often exploiting heterosis, hybrid vigor, which enhances traits like fertility, survivability, and growth rate in the F1 generation. Modern biotechnology has further enriched breeding programs. Artificial insemination (AI) facilitates widespread use of genetically superior males. Embryo transfer technology (ETT) and in vitro fertilization (IVF) accelerate multiplication of elite females. Somatic cell nuclear transfer (SCNT) or cloning allows exact replication of elite genotypes. However, cloning suffers from ethical concerns, low success rates, and developmental anomalies. CRISPR (Cas9) gene editing now offers precise manipulation of the animal genome. It enables the insertion, deletion, or replacement of specific DNA sequences, allowing the creation of transgenic or gene-edited animals with improved traits. For example, pigs resistant to PRRS virus have been developed using CRISPR. Similarly, culled cattle have been engineered to eliminate the need for dehorning. For sustainable animal breeding, it's essential to balance genetic gain with conservation of biodiversity. Indigenous breeds often possess superior adaptability, disease resistance, and lower input requirements. Programs like Rashtriya Gokul Mission in India aim to conserve and improve native breeds using genomic and reproductive technologies. In conclusion, animal breeding today is a sophisticated interplay of classical selection, quantitative genetics, reproductive technologies, and molecular genomics. CSIR-NET aspirants must understand the principles of heritability, selection response, QTLs, MAS, GS, and gene editing tools to tackle applied questions effectively. Previous year CSIR-NET questions with answers, CSIR-NET Deck 2016. Which of the following best explains the advantage of marker-assisted selection over traditional phenotypic selection? A MAS is only effective for traits with high heritability. B. MAS requires no knowledge of gene function. C. MAS allows selection before trait expression. D. MAS works only in plants. Answer: C. MAS allows selection before trait expression. CSIR-NET June 2018. In animal breeding, which strategy is most suitable for improvement of traits with low heritability? A. Phenotypic selection. B. Crossbreeding. C. Marker-assisted selection. D. Random mating. Answer: C. Marker-assisted selection. CSIR-NET Deck 2022. Which gene is used as a marker in MAS to improve meat quality in pigs? A. FECB. B. PRNP circa RYR1 DDGAT1. Answer: C. RYR1. Let me know the next topic or if you want me to continue with the plant breeding lecture including MAS. You said. Marker, assisted selection. ChatGPT said. Here's the CSIR-NET JRF Life Sciences high-level lecture on marker assisted selection MAS. Full length, highly exam focused, structured as a single paragraph, includes previous year questions with answers in years. Highly exam focused, structured as a single paragraph, includes previous year questions with answers in years. Marker assisted selection MAS is a powerful technique in modern genetics and breeding that uses molecular markers to identify individuals carrying desirable traits for selection and propagation. Unlike conventional breeding, which relies solely on phenotypic traits, MAS allows the selection of traits at the DNA level, even before they are expressed. This is particularly valuable for traits with low heritability, traits expressed late in life like reproductive performance or yield, or those that are difficult, expensive, or destructive to measure, e.g., disease resistance, fiber quality. MAS involves the use of molecular markers such as SSRs, symbol sequence repeats, SNPs, single nucleotide polymorphisms, RFLPs, restriction fragment length polymorphisms, and AFLPs, amplified fragment length polymorphisms, that are tightly linked to genes or quantitative trait loci, QTLs, controlling traits of interest. The process ends with QTL mapping, where markers closely linked to desired traits are identified using linkage mapping or association studies in a population with known phenotypes. Once these markers are validated, they are used for genotyping individuals in breeding populations. Individuals carrying favorable alleles are then selected, thereby increasing the efficiency and accuracy of breeding programs. MAS has been extensively applied in both plant and animal breeding. In plants, it is used to incorporate traits such as disease resistance, e.g., bacterial blight in rice via AXA21 gene, drought tolerance, yield components, and nutritional improvements, e.g., high provitamin content in maize via CRTRB1 gene. In animals, it helps in selecting against genetic disorders, e.g., blood in cattle, or enhancing traits like growth rate or carcass quality. The key advantages of MAS are that it reduces the breeding cycle duration, increases selection efficiency, and facilitates gene pyramiding, where multiple favorable genes for the same or different traits are stacked into a single genotype. Unlike phenotypic selection, MAS is unaffected by environmental conditions and can be applied to seedlings or juvenile stages, which significantly accelerates the process. One limitation of MAS, however, is its reduced efficacy for polygenic traits, which are influenced by multiple genes with small effects. This has led to the development of genomic selection (GS), which uses genome-wide marker data to predict genetic merit through statistical models, especially useful in traits with a complex genetic basis. In India, several breeding programs have successfully integrated MAS, such as the development of improved Pusa basmati 1 with resistance to bacterial blight and Swarna sub 1 rice submergence tolerance. For success in MAS, it is critical that markers be tightly linked to the gene QTL, ideally within 5 cm, have high polymorphism, and be reproducible across generations and genetic backgrounds. Thus, MAS represents a marriage of classical breeding with molecular biology, offering a precise, rapid, and cost-effective route for genetic improvement in agriculture and animal husbandry, and is a frequently tested concept in CSIR-NET due to its strong conceptual and applied significance. Previous year CSIR-NET questions with answers, CSIR-NET Deck 2016. Which of the following best explains the advantage of marker-assisted selection over traditional phenotypic selection? A MAS is only effective for traits with high heritability. B. MAS requires no knowledge of gene function. C. MAS allows selection before trait expression. D. MAS works only in plants. Answer: C. MAS allows selection before trait expression. CSIR-NET June 2018. In a rice breeding program, a line with resistance to bacterial blight is crossed with a high-yielding susceptible line. To introgress the resistance trait, which method is ideal to identify progenies carrying the resistance gene? A. Backcrossing followed by phenotypic selection. B. Hybridization without selection. C. Marker-assisted selection using gene-specific markers. D. Pure line selection. Answer: C. Marker-assisted selection using gene-specific markers. CSIR-NET Deck 2022. Which of the following is not an advantage of marker-assisted selection? A. It allows gene pyramiding. B. It accelerates breeding cycle. C. It is independent of environmental conditions. D. It always performs better than phenotypic selection in polygenic traits. Answer: D. It always performs better than phenotypic selection in polygenic traits. Let me know the next topic you'd like to continue with. You said, bioremediation. ChatGPT said. Here is your CSIR-NET JRF Life Sciences High Quality, full length lecture on bioremediation. Highly exam-oriented, detailed from base to advanced level, note tables, paragraph format, includes previous year questions and answers with years. Bioremediation is the process of using living organisms, primarily microbes and plants, to degrade, detoxify, or remove environmental contaminants such as heavy metals, hydrocarbons, pesticides, solvents, and industrial waste from soil, water, and air. It is a sustainable and eco-friendly alternative to chemical or physical methods of pollution control and is a critical topic under applied biology for the CSIR-NET Life Sciences syllabus. The organisms used for bioremediation, including bacteria e.g., Pseudomonas, Bacillus, Deinococcus fungi e.g., Aspergillus, phanerochaete algae, and even genetically engineered microbes, possess metabolic capabilities to utilize pollutants as energy or carbon sources, or convert them into less harmful forms through enzymatic activity. Bioremediation processes can be classified based on environment into in situ, on-site, and ex situ, off-site techniques. In situ methods like bioventing, biosparging, and natural attenuation are less disruptive and cost-effective, while ex situ techniques like bioreactors or land farming involve excavation and treatment elsewhere but offer greater control. A key mechanism in microbial bioremediation is metabolic transformation, where microbes enzymatically break down organic pollutants. For example, Pseudomonas petita degrades toluene and xylene through dioxygenase pathways. In co-metabolism, microbes degrade contaminants incidentally while metabolizing another compound. This is common in the degradation of trichloroethylene (TCE) by methane-oxidizing bacteria. Bioaugmentation involves introducing specific strains of microorganisms with desired degradation capabilities into contaminated environments to enhance remediation. In contrast, biostimulation adds nutrients or electron acceptors like nitrate, phosphate, or oxygen to stimulate the native microbial community. Heavy metal bioremediation often involves biosorption, where metals like lead, mercury, cadmium, or chromium bind to the microbial cell surface through functional groups e.g., carboxyl, amino, phosphate, or bioaccumulation, where they are transported into the cell. Some bacteria also chemically reduce toxic metals. For example, Geobacter sulfureticens reduces soluble U (B) to insoluble U (IV). Phytoremediation, another form of bioremediation, uses plants such as Brassica uintia, Helianthus annuus, or Populus species to absorb, accumulate, and degrade pollutants. Its types include phytoextraction, metal uptake, phytodegradation, enzymatic breakdown, phytostabilization, immobilization of contaminants, and rhizofiltration, root-based absorption from water. Modern bioremediation employs genetically engineered microorganisms (GEMs) to enhance pollutant degradation. One classic example is Superbug, Pseudomonas developed by Dr. Ananda Chakrabarty, capable of degrading multiple hydrocarbons in oil spills. Another is the engineering of E. coli to express meroperon, enabling detoxification of mercury. Use of GEMs in the field requires strict regulatory monitoring due to potential biosafety and ecological concerns. Recent advancements include the use of synthetic biology, metagenomics, and nanobiotechnology to design microbial consortia or enzyme systems that are tailored for specific contaminants and resistant to harsh conditions. Bioremediation is influenced by environmental parameters like pH, temperature, oxygen availability, salinity, and the bioavailability of the contaminant. Contaminants embedded in non-aqueous phase liquids (NaPLs) or bound to sediments are often poorly bioavailable, limiting biodegradation. Surfactants, either synthetic or biosurfactants produced by microbes, are sometimes used to enhance contaminant solubilization. Monitoring bioremediation progress involves assessing microbial population dynamics, contaminant disappearance, metabolic byproducts, and gene expression profiles using molecular tools such as qPCR, metagenomics, and fluorescent probes. Bioremediation has been successfully applied in many real-world contexts. Oil spills e.g., Exxon Valdez, pesticide cleanup e.g., organochlorines, industrial effluent treatment e.g., textile dyes, and restoration of heavy metal contaminated mining sites. Despite its effectiveness, bioremediation faces challenges such as incomplete degradation, accumulation of toxic intermediates, and the slow rate of microbial action under extreme environmental conditions. Regulatory and public acceptance also affect its implementation. However, with ongoing innovations in microbial engineering, systems biology, and environmental monitoring, bioremediation remains a cornerstone of sustainable pollution management and a frequently tested concept in CSIRO-NET. Previous year CSIRO-NET questions with answers, CSIRO-NET Deck 2017. Which of the following microbial genera is commonly associated with degradation of aromatic hydrocarbons in polluted environments? A. Rhizobium. B. Clostridium. C. Pseudomonas. D. Streptococcus. Answer, C. Pseudomonas. CSIRO-NET June 2020. Phytoremediation of heavy metals primarily involves which of the following mechanisms? A. Hydrolysis of pollutants. B. Uptake and accumulation in plant tissues. C. Bacterial fermentation and rhizosphere. D. Evaporation through leaf stomata. Answer, B. Uptake and accumulation in plant tissues. CSIRO-NET Deck 2019. The function of biosurfactants in bioremediation is to A. Kill non-degrading bacteria. B. Chelate heavy metals. C. Increase pollutant solubility and bioavailability. D. Enhance plant root growth. Answer, C. Increase pollutant solubility and bioavailability. Let me know the next topic you'd like me to cover with the same level of detail. You said, Phytoremediation. ChatGPT said. Here is your CSIRO-NET JRF Life Sciences in-depth, high-standard lecture on phytoremediation. Extensive, exam-focused, note tables, structured in paragraph form, includes previous year CSIRO-NET questions with answers in years. Phytoremediation is a cost-effective, environment-friendly biotechnological approach that uses green plants and their associated microbiota to detoxify, extract, immobilize, or degrade pollutants from soil, water, and air. It is a subset of bioremediation and is particularly effective for removing heavy metals, radionuclides, pesticides, petroleum hydrocarbons, and excessive nutrients from contaminated sites. This technique leverages natural plant processes like uptake, metabolism, transpiration, and root microbe interactions to mitigate contamination, and has emerged as a sustainable alternative to physical and chemical remediation techniques. The key to successful phytoremediation lies in the selection of appropriate plant species, often referred to as hyperaccumulators, which are capable of tolerating and accumulating large amounts of specific contaminants without phytotoxic effects. Examples include Brassica uintia, Indian mustard, for lead and chromium, Helianthus annuus, sunflower, for uranium, Thalaspi caryolescens for zinc and cadmium, and Terry spitata for arsenic. Phytoremediation comprises several strategies based on the type of contaminant and its fate in the plant. Phytoextraction, phytoaccumulation, is the uptake and concentration of contaminants, usually heavy metals, in the harvestable parts of plants. The success of this strategy depends on the plant's biomass and its ability to translocate metals from roots to shoots. After harvesting, the biomass can be incinerated or processed to recover valuable metals, phytomining. Phytostabilization involves the immobilization of contaminants in the root zone, reducing their bioavailability and leaching into groundwater. This method is ideal for preventing the spread of toxic metals like cadmium, zinc, or lead in mine tailings or industrial waste sites. Phytodegradation, phytotransformation, refers to the uptake and enzymatic breakdown of organic pollutants such as herbicides, pesticides, explosives, and solvents within plant tissues using enzymes like peroxidases, lacases, or nitroreductases. Plants like Populus species and Cannabis sativa are effective in degrading organic contaminants. Phytovolatilization involves the uptake of volatile contaminants like mercury or selenium and their release into the atmosphere in a less harmful form. While this technique reduces soil or water contamination, it transfers the pollutant to the air and is therefore controversial for highly toxic elements. Rhizofiltration, another common method, uses plant roots to absorb, concentrate, and precipitate metals from aqueous waste, typically employed for wastewater or groundwater cleanup. Plants like Limnobium, Eichhornia, and Vetiver are used in such systems. The success of phytoremediation depends on factors such as plant root depth, growth rate, tolerance to pollutants, and rhizospheric interactions. The rhizosphere, the soil region influenced by root secretions, plays a crucial role, as it hosts microbial populations that contribute to degradation, mobilization, or immobilization of contaminants. Plant microbe consortia, including nitrogen-fixing bacteria, mycorrhizae, and endophytes, can significantly enhance phytoremediation efficiency. For instance, arbuscular mycorrhizal fungi improve plant growth under metal stress and increase uptake of phosphorus and micronutrients. Phytoremediation can be further enhanced by genetic engineering, where plants are modified to express metal-binding proteins like metallothioneins, phytochelatins, or foreign enzymes capable of breaking down organic toxins. Transgenic Arabidopsis expressing bacterial mercuric reductase (MERA) and organomercurial lyase (MERP) genes have shown enhanced mercury volatilization. Despite its potential, phytoremediation has limitations. It is slower than conventional methods, often restricted to surface contamination, and is ineffective in highly contaminated or toxic soils where plant survival is compromised. However, with increasing pollution from mining, industry, and agriculture, phytoremediation is gaining recognition for its ecological sustainability and low operational costs. It also has aesthetic and land restoration value and is being incorporated into green technologies for urban landscaping, constructed wetlands, and biomining. In India, phytoremediation is being explored for cleaning fly ash dumps, tannery effluent zones, and arsenic-contaminated regions in West Bengal and Assam. The method is a regular topic in CSIRO-NET Life Sciences due to its integration of molecular biology, environmental science, and plant biotechnology, making it essential for applied research in real-world environmental management. Previous year CSIRO-NET questions with answers, CSIRO-NET June 2019. Which of the following plant species is known for its capacity to hyperaccumulate arsenic from soil? A. Brassica uintia. B. Terry spitata. C. Arabidopsis thaliana. D. Oryza sativa. Answer, B. Terry spitata. CSIRO-NET Deck 2016. What is the primary mechanism in phytostabilization? A. Volatilization of contaminants through stomata. B. Accumulation of metals in shoots. C. Degradation of pollutants inside the leaf cells. D. Immobilization of contaminants in the rhizosphere. Answer, D. Immobilization of contaminants in the rhizosphere. CSIRO-NET Deck 2020. Which of the following is not typically considered a phytoremediation strategy? A. Phytomining. B. Rhizofiltration. C. Phytodegradation. D. Biosparging. Answer, D. Biosparging. Let me know the next topic you'd like me to cover with the same depth and structure. You said, biosensors. ChatGPT said. Here is your CSIRO-NET JRF Life Sciences High Standard, detailed lecture on biosensors. CSIR net focused, paragraph format, maximum word limit, no tables, includes previous year questions with answers in year. Biosensors are analytical devices that combine a biological recognition element with a physiochemical transducer to detect and quantify analytes with high specificity and sensitivity. These devices are widely used in medical diagnostics, environmental monitoring, food safety, and bioprocess control. A biosensor consists of three primary components. 1. A biorecognition element, such as enzymes, antibodies, nucleic acids, whole cells, or receptors that selectively interacts with the target analyte. 2. A transducer that converts the biological interaction into a measurable signal, such as optical, electrochemical, piezoelectric, or thermal. And 3. A signal processing system that amplifies, interprets, and displays the signal. The central principle of biosensors lies in their ability to transduce a biochemical event into a quantifiable output, often in real time, allowing for rapid and on-site detection. Biosensors are classified based on the type of transducer or biological element employed. Electrochemical biosensors are the most commonly used and detect changes in current, amperometric, potential, potentiometric, or impedance conductometric in response to biochemical reactions. These are widely applied in glucose monitoring using glucose oxidase. Optical biosensors use light absorption, fluorescence, or surface plasmon resonance (SPR) to detect interactions such as antigen-antibody binding or DNA hybridization. SPR-based biosensors have become indispensable in studying molecular interactions without the need for labeling. Thermal biosensors, or calorimetric sensors, detect heat changes during enzymatic reactions, while piezoelectric biosensors rely on changes in mass or acoustic waves upon analyte binding, typically using quartz crystal microbalances. The choice of biosensor depends on the nature of the analyte, required sensitivity, and field of application. The biological recognition elements determine the specificity of biosensors. Enzyme-based biosensors catalyze substrate conversion, and the product or associated change, pH, temperature, etc., is measured. For example, urease-based sensors detect urea levels via the pH change produced by ammonia formation. Antibody-based biosensors, or immunosensors, rely on antigen-antibody binding, and are used in the detection of disease markers like HIV proteins or cancer antigens. Nucleic acid biosensors detect complementary DNA or RNA sequences, with applications in pathogen detection and gene expression profiling. Aptamer-based biosensors, using short synthetic oligonucleotides, offer high affinity and specificity, especially for small molecules, and are gaining popularity due to their stability and reusability. Whole cell biosensors use metabolically active cells that respond to toxins, pollutants, or metabolic changes. These are used in environmental and toxicological monitoring. Biosensor design and signal amplification have advanced with nanotechnology and synthetic biology. Nanomaterials such as gold nanoparticles, carbon nanotubes, graphene, and quantum dots enhance the sensitivity and stability of biosensors by increasing surface area and electron transfer rates. For example, gold nanoparticle modified electrodes enhance electron transfer in electrochemical biosensors, while quantum dot labeled antibodies improve fluorescence detection. Additionally, synthetic biology enables the construction of genetically engineered microbes or cell-free systems that act as programmable biosensing platforms. These advances have led to the development of lab-on-a-chip devices and point-of-care diagnostics that integrate biosensors into compact, user-friendly systems suitable for clinical and field use. Applications of biosensors span diverse domains. In medical diagnostics, biosensors detect biomarkers for diseases like diabetes, glucose sensors, cancer, tumor markers, cardiac conditions, troponins, and infectious diseases, antigen, antibody detection. In environmental monitoring, biosensors detect pollutants such as heavy metals, pesticides, and pathogens in water and soil. For instance, genetically engineered bacterial biosensors expressing GFP, green fluorescent protein, in response to mercury are used for toxicity screening. In agriculture, biosensors are employed to monitor crop pathogens, soil nutrient levels, and pesticide residues. Food industry applications include detecting contaminants like aflatoxins, foodborne pathogens e.g., salmonella, and spoilage indicators. Industrially, biosensors are used in bioprocess monitoring, ensuring real-time control over fermentation parameters such as glucose, lactate, or oxygen levels. Despite their wide applications, biosensors face challenges including stability, reproducibility, shelf life, and nonspecific binding, especially under real-world conditions. Advances in material science, immobilization techniques, and miniaturization are overcoming these limitations. One major trend is the integration of biosensors with smartphones and wearable devices for continuous health monitoring. Glucose sensors embedded in wearable patches or contact lenses are examples of next-generation biosensing technologies. In the context of CSIR Net Life Sciences, biosensors are often linked to applied biology, immunology, enzymology, nanobiotechnology, and environmental biology. Questions test understanding of biosensor types, mechanisms of detection, enzyme antibody-based applications, and current trends in biosensing technology. Mastery of these concepts is essential for both conceptual and application-based questions in the exam. Previous year CSIR Net questions with answers, CSIR Net December 2015. Which of the following components of a biosensor is responsible for converting the biological signal into an electrical signal? A. Bioreceptor. B. Transducer. C. Amplifier. D. Processor. Answer, B. Transducer. CSIR Net June 2018. In an amperometric biosensor used for glucose detection, which enzyme is typically used? A. Glucose isomerase. B. Glucose oxidase. C. Hexokinase. D. Invertase. Answer, B. Glucose oxidase. CSIR Net Deck 2020. Which of the following is not a type of biosensor transduction method? A. Optical. B. Thermal. C. Magnetic resonance imaging. D. Piezoelectric. Answer, C. Magnetic resonance imaging. Let me know your next topic for CSIR Net Life Sciences, and I'll continue in the same style with maximum quality and depth.